All right, guys, here we are. We are still on cellular respiration. This podcast, though, is taking us from glycolysis into the citric acid cycle, which is step 2 of the cellular respiration process. So if we take a look at the overview again, this is where we are. We just finished up with glycolysis, where our 6-carbon glucose was broken down to 2 3-carbon pyruvates. And those pyruvates now are going to move into what we call the citric acid cycle. So let's take a brief look at glycolysis again to see what's happening. So again, with glycolysis, we had energy input and output. Why did we have energy input? Well, we always need energy to break that bond. So we started off with the 6-carbon glucose. ATP came in, broke that bond, and so now we are left with 2 3-carbon molecules. In this energy payoff phase, though, what happens next is, remember I said you're more than welcome to get your textbook out and study your chemistry heart out as much as you want. But all you need to know for AP Bio is this, that this is a series of reactions that are catalyzed by enzymes. And what's going to happen is we're going to get 4 ATP produced due to substrate level phosphorylation. And we're going to get 2 NADH. Remember, NAD is our electron carrier. And so NAD has now been reduced because it's gained electrons. And it's now NADH. And we get these 2 pyruvate. Now we know that ATP is going to be used for cellular work. And we've talked a little bit about, that NADH, that's going to move on to oxidative phosphorylation, where we're going to go to the electron transport chain. But what the heck happens to these 2 pyruvate, to these 2 3-carbon molecules? Well, that's what we're going to look at today. So these 2 3-carbon molecules are going to enter the citric acid cycle. And what is that? Well, its function is to simply oxidize pyruvic acid, or that pyruvate, to carbon dioxide. In the same time, it's going to produce NADH and FADH2, and the location that all this occurs in is the mitochondrial matrix. So let's take a quick overview of what's going on here. So we start with pyruvate. Now remember, we're just going to look at 1 pyruvate here, but remember there's 2 of these because we had 1 glucose, and that 1 glucose broke into 2 pyruvates. And what we can see happen here is, I like to call this like the ball and chain reaction. For whatever reason, this reaction always reminds me of a wrecking ball. So here's my chain, and then the actual cycle, which is the citric acid cycle, or otherwise sometimes referred to as Krebs after the scientist. Krebs cycle is the ball here. Okay, so this is kind of an overview of what we're going to be covering. So what are the requirements of the citric acid cycle? Well, in order for this cycle to run, pyruvate must be present, coenzyme A must be present. We need to have some electron carriers in the form of NAD. We need to have ADP because we're going to do some substrate level phosphorylation here. And we also need a new electron carrier, which is called FAD. Now, you don't need, just know it's an electron carrier. Okay? Now remember, again, I'm going to say it again and again, 2 pyruvate enter the citric acid cycle per 1 glucose. So this is what we need in order for 1 pyruvate to go through. So obviously we're going to have to double in order to get all of the both pyruvates through. So what are the products of the citric acid cycle? Well, the first product produced from each pyruvate. So again, we're going to double this list to show what is produced from 1 glucose. From each pyruvate, I'm going to get 3 carbon dioxides. I'm going to get an acetyl-CoA. I'm going to get 4 NADH, 1 ATP, and 1 FADH2. Okay, again, to see what is produced from a glucose molecule, we simply double this list. Okay, so those are our end products. Well, let's talk about how we get there. When we look at the formation of acetyl-CoA, that's the first step. So remember I told you I see the citric acid as this ball and chain. And so the chain part, this is where we take the pyruvate and it enters the mitochondria. Now there's a transport protein here. And the purpose of that transport protein is because pyruvate is a charged molecule, so it moves in through active transport. Okay, we're moving from low to high here. And so pyruvate enters through that transport protein. Now the first thing that's going to happen is check out this carboxyl group here. You see that COO guy? Well, he has already been oxidized. He has very, very little energy left. So the first thing that's going to happen is that carboxyl group is going to be released as carbon dioxide. So we see CO2 coming off. The next thing that's going to happen is NAD is going to come in, and it is going to become reduced, grab some electrons, and form NADH. Okay, and so we're going to extract some of those electrons from this pyruvate molecule. Now, once CO2 has left pyruvate, it has now become a molecule called acetate because we only have 2 carbons. So then NAD comes in, grabs some electrons from that acetate molecule, and it's going to take that to the electron transport chain. And then coenzyme A is going to come in and attach to that acetate. And so now we have this 2-carbon molecule called acetyl coenzyme A. Okay? Again, this is happening in the mitochondria right here. Right? And the only way that pyruvate is going to enter into the mitochondria is if oxygen is present. Now you can see we're not using oxygen yet. But it will only do this if oxygen is present, because the end result requires oxygen to occur. And we'll talk about what happens what if there is no oxygen present? What's pyruvate going to do? Well, we'll talk about what pyruvate does if oxygen is not present. But if it is present, that pyruvate is going to go through that transport protein via active transport. It is going to lose its carboxyl group, so when that carboxyl group is removed as carbon dioxide, it has now become acetate. We're right here. NAD comes, takes a couple of electrons from that acetate, and now that acetate is willing to bond to coenzyme A. Now remember, coenzyme is simply an organic non-protein molecule that helps aid enzymes work. That's it. So you should kind of know, well, if coenzyme's coming in, that means we're going to be going through a lot of reactions that are going to be utilizing those enzymes. So let's take a look at that. Okay, whew. Look at this. Guess what? You don't need to memorize this, but let's take a look so we can appreciate what's happening. Acetyl-CoA, now here, is going to come in, and we have oxaloacetate. And once with oxaloacetate, we're going to enter into this 8-step process. Now notice here that the carbons are in red. Everybody see that? See my carbons that are in red and how it matched up with these carbons that are in blue. If they're in red, it means that they are reduced. Okay? If they're in blue, it means that they are already oxidized, which means that they are very, very low energy. So we want to take these 2 carbons and end up oxidizing them through the citric acid cycle. So how do we do that? Well, through a series of reactions. Remember, each arrow is going to be a reaction, and we have an enzyme at each spot. One, we can see water is formed. Okay. We here can see NAD is going to come in. It's going to grab some electrons, become reduced, and form NADH. And again, we're going to see NAD come in here. Oh, I forgot. There's a carbon dioxide that was released there. That means a carboxyl group was released. okay? And you can follow the chemicals that are, I'm so sorry. You can follow the compounds that are going here. I think I'm getting old. It's hard for me to see these little things. So NAD, again, is going to come in down here, grab some electrons, forms NADH. And while we're doing is reactant becomes the product, right? And then that product is a reactant for the next reaction. So it's just a cycle that goes through. This whole cycle where we are simply producing NADH, ATP, and FADH2. At the same time, we are releasing waste products in the form of carbon dioxide and water. Now check this out. You can see here, seriously, it didn't just do that. All right, guys, I'm sorry about that. So here we go. You can see here when those red carbons, okay, what's going on when they become completely oxidized, because now we're up here to blue. So this is my favorite part. NAD comes in, takes those remaining electrons, and converts it to oxaloacetate. Guess what? Now we have oxaloacetate again to grab on to the next acetyl-CoA, and then the reaction can go again. So for each glucose molecule that goes through cellular respiration, the citric acid cycle is going to turn twice. Okay? So what do you need to know? I like this one because, boy, keeps it short and sweet, where you can see exactly what is released and what is produced. Remember, on the citric acid cycle, the ATPs, those 2 per glucose molecule, remember, are produced directly in the citric acid cycle, and in glycolysis, they're produced by substrate-level phosphorylation. And what is that? That's when an enzyme transfers a phosphate group from a substrate molecule to ADP. This is direct. So when this happened in glycolysis, we could see our substrate here that had a phosphate. The enzyme joined that phosphate, phosphorylated ADP, and henceforth produced ATP, and we got the product pyruvate, which was then going to head into our citric acid cycle. This is an example of substrate-level phosphorylation, where ADP picks up a phosphate from the substrate. Okay, doesn't produce a whole heck of a lot. So final comments here about citric acid cycle. It produces most of the cell's energy in the form of NADH and FADH2, which are electron carriers, and now we'll go to the electron transport chain. This does not require oxygen. Their oxygen has not been utilized yet. Okay, and we're going to see where oxygen fits into the picture with our electron transport chain. But in the end, what we get is a production of only per pyruvate. Okay, we're only going to get our 1 ATP. We simply get our 4 NADH, and we get our 1 FADH2. Okay, so per glucose, we're getting 2 ATP, 8 NADH, and 2 FADH2. And in the next podcast, we're going to look at the electron transport chain and what do we do now with all these NADHs and FADH2 that are carrying that energy that we want to convert into ATP for cellular work. Until then, see you later.